Hi, welcome to Neuroverse, a podcast hosted by Carolina
and Clara, where we discuss all matters from neuroscience to philosophy and beyond.
Hi, welcome to today's episode on antibiotic resistance. I chose this topic because I think antibiotic resistance is a really important topic that sometimes is not discussed as often, and I think it's good to shine a light on how bacteria have been evolving and becoming more resistant to antibiotics, but also how it would be good to have a little bit more of a spotlight on having more research and funding going into this area.
Yeah, and I think it became clear to me through researching this topic that there is still so much work to be done. Like, there's still so much not understood about how this resistance comes about and how we can stop it, really, which is the important question.
Yeah. So antibiotic resistance is when bacteria change and become resistant to antibiotics, making it harder to treat bacterial infections. Just as an overview, this happens through genetic changes, so bacteria can mutate and develop resistance to antibiotics. This can also happen through sharing genetic material. Bacteria can transfer resistant DNA to other bacteria, and also through the overuse of antibiotics. What it means is that infections will be harder to treat, alternative treatment may be more expensive or toxic, and resistant bacteria can spread to other people and be more deadly.
Yeah, and it's crazy the impact that antibiotic resistance has on human health. I read that the Center for Disease Control and Prevention, the CDC in the US, have declared 15 antibiotic-resistant human pathogens as urgent or serious threats to human health, estimating that 2 million people within the US alone are sick every year due to antibiotic-resistant infections.
Yeah, that's a really big number. And The Lancet published this study that it estimated that by 2050, around 39 million people will die from antimicrobial resistance, which is around 2 million people per year, which is an enormous number.
Yeah, wow.
Yeah. And I also wanted to point out that even though this episode is about antibiotic resistance, that antimicrobial resistance as a whole is really important. So fungi have also become resistant and viruses are also affected. But for today's episode, we're just going to focus on bacteria because they're already so diverse and complex as it is.
Mm-hmm. And yeah, I discovered that antibiotic resistance is actually something that has been around on Earth for much longer than I would have thought. And most antibiotics that are used in the clinic or for agricultural uses, which are the two major uses of antibiotics, are derived from a group of soil-dwelling bacteria called actinomycetes that naturally produce these metabolites, which are the antibiotics, such as streptomycin, tetracycline, and other commonly known ones. And these organisms must therefore be themselves resistant to the antibiotics that they produce. And so these may have been the original sources for many antibiotic-resistant genes today. But interestingly, there have been several studies looking at really ancient samples from permafrost or isolated caves or ancient human or animal specimens that existed long before the use of antibiotics in the clinic and have found resistance genes. For example, one study showed the presence of genes resistant to tetracycline, beta-lactam, glycopeptide antibiotics in samples from 30,000-year-old permafrost, which also contained DNA belonging to woolly mammoths and other species of that period, which I think helps to kind of visualize the scene a bit better.
So cool.
And another study looked at the gut microbiome of a Andean mummy from Cusco, Peru, dating back to 980 to 1170 AD, which was found to harbor genes that were homologous to some antibiotic resistance genes, suggesting that the human microbiome has been a source of resistance genes, even in the absence of modern antibiotic availability. However, that is not to say that this crisis of antibiotic resistance is not being accelerated by the use of antibiotics. It definitely has been. But on top of the already naturally occurring antibiotic resistance genes that have, been propagated across the world and are continuing to, it is a major problem that I think we still need to understand how to overcome.
Yeah. Yeah, it's so amazing to hear about how in nature these antibiotic resistance genes can occur. And yeah, it's kind of scary to think about how more and more ice is melting, and now all of these things that have been frozen in time are just thawing
and being released.
Yeah. So I want to talk a little bit about the mechanisms in how bacteria actually fight antibiotics, because it's kind of cool. It almost seems intelligent. I know it's just evolution, but it's so detailed that, it's very interesting. So bacteria can inactivate antibiotics by degrading or modifying the antibiotic molecule, and they do this through enzymes and enzymatic degradation. And specifically, for those that are interested, they hydrolyze the functional group of the antibiotic. But yeah, basically they just degrade the antibiotic, rendering it ineffective. Another method is they do target site alteration.
Wait, sorry. So do they produce enzymes that target the antibiotics?
Yeah.
And they evolve the genes to produce these enzymes?
Yeah. They also do target site alteration, which means that they alter their site which the antibiotics target. So they alter their site through genetic mutations and also through enzymatic alterations of the binding site. So they alter their own site where the antibiotics target and bind to. And they also do something called a target bypass, which is when they create a new protein for the antibiotic to target, and therefore making it ineffective by leaving the actual target site unaffected, which is crazy how they are able to evolve to be able to do such a complex thing. And essentially this new target protein has the same metabolic capacity as the original target site, so it kind of makes the antibiotic waste its energy. And they also prevent the antibiotics from actually entering the bacteria by preventing them to cross their cell wall and through the transmembrane proteins that they have, by decreasing the influx. So they downregulate their porins, which makes it harder for the antibiotics to get through. And also they do something called the efflux, where they basically flush out the antibiotics out of their cell wall. So in every direction.
Yeah, I was thinking they're really covering all bases.
Yeah.
It's like they have a mechanism for every possible entry or effect.
Mm-hmm. Yeah.
Yeah, and another unique thing about bacteria is their ability to then transfer these transfer these genes that give them the ability to fight off the antibiotics across bacteria. And this can be through two ways. One is they can acquire genes from their parent microorganisms during division, which is known as vertical gene transfer. But they can also acquire genes from the community at large, which is known as horizontal gene transfer.
Yeah, horizontal gene transfer is so interesting because they can do it in 3 different ways. The first one is through transformation, where they uptake free DNA from dead bacteria. They also do transduction, which is where a transfer of genes by bacteriophages occurs. So these bacteriophages are viruses that infect bacteria. And thirdly, through conjugation, which is simply when bacteria come into direct contact with other bacteria.
Yeah, it's crazy. It's like if humans didn't just pass on their genes to their offspring, but you could like give your friends your genes.
Yeah.
That's
That would be kind
essentially
of
what it
nice.
is. Yeah, you could just choose your traits.
Yeah. And another mechanism that these bacteria employ is when they come into direct contact with an antibiotic, they upregulate the genes that do things such as upregulate efflux pumps to pump out the bacteria. So this allows them to be really conserved with their bacteria and only employ these defense mechanisms when is actually necessary.
Which I guess maybe why then increasing exposure of bacteria in the world to antibiotics in general through an increased use of antibiotics can also enhance their development of these mechanisms against the antibiotics.
Yeah, definitely. And as mentioned, these bacterial enzymes evolve to degrade or modify antibiotics. And essentially the scary thing is that with the fact that through antibiotic resistance, penicillin and many other commonly used antibiotics become ineffective. And that kind of brings us back to the Victorian era. So something like a simple bacterial infection is now maybe actually life-threatening.
Yeah. And I think what's crazy is also that the majority of anti-resistance genes that we know of have an unknown origin, maybe because they originate from a species that we haven't yet sequenced. But also, as we've discussed, there's a whole host of different ways that they could be developed, namely either evolutionarily, which would lead to the introduction of a whole new resistance factor gene, or that a transmission event which spreads already widespread resistance strains to more bacterial species. And so it's difficult to tear apart whether these mutations have come from an evolutionary or a transmission event, essentially.
Yeah. And especially because bacteria can acquire, as mentioned, the horizontal gene transfer from dead or alive or viruses.
Yeah.
It's like the options are endless. No wonder it's really difficult to find effective measures to combat antibiotic resistance when they're ever changing.
Yeah, I also found out that when studying antibiotic resistance genes, people normally look at like clinical samples because it's easier and more because it's easier and less variable maybe than say environmental samples. And so when looking at environmental samples as well, you don't really know if you're just looking at free bacteria free DNA, or if you're looking at like bacterial DNA.
And so, as you mentioned earlier, there are many sources that is driving antibiotic resistance, so from the environment clinically and also through agriculture. And there's been studies that have shown that approximately 80% of total consumption of antibiotics is from the agricultural sector.
Yeah. Oh my gosh.
Yeah.
Wow.
It's a big number. But there have been governmental and organizational efforts to combat this. So the WHO recommended for farmers to stop using antibiotics routinely on healthy animals, because that's the other thing. Antibiotics are meant to just be used when animals or people are unhealthy or sick, but they're using them in perfectly healthy animals to prevent disease. Yeah. And in 2022, the EU banned the use of antibiotics in agriculture in this measure, where they're using it as a preventative rather than as a curative, which is good, at least. And the UK has similar regulations, but they're not as strict as the EU. So I'm not sure what that means, but I think that they're not as strict in enforcing this, so that may still be happening in the UK. But they have cracked down on the amount that agriculture has been using antibiotics.
Well, I was reading about how, like, the global burden of antibiotic resistance, and I was interested in finding what the global disparities were, because of course this is a global crisis. Like if countries don't work together, then, you know, even if certain countries have regulations that decrease the spread of these resistance genes, if they're still being developed somewhere else in the world, it will eventually get to us. And so anyways, I was interested in that. And it's related to agriculture because unfortunately, so first of all, we'll maybe talk about this more. It's not the most pleasant topic, but resistant bacteria spread a lot through feces. Also because fecal bacteria are known to carry certain elements that can help to relocate genes between cells, and so they may also be a
medium.
Yeah, like a ideal medium for resistance gene transfer to occur. But because of this, antibiotic resistance levels are very much related to general hygiene. And what I found surprising was that one study did a correlative analysis and found that overall sanitation and waste infrastructure was actually a better predictor of national burdens of antibiotic resistance than the reported use of antibiotics.
Wow.
So overall sanitation and waste management that would affect the spread of the antibiotic resistance genes is actually more important than controlling the use of the antibiotics. And then related to agriculture, in a lot of lower income countries, agriculture is a large industry and they are forced to use methods like preventative antibiotics, as you explained, in order to just have like really high volumes of animals being produced, and are supplying, you know, other countries with that meat, for example. And so it's actually a huge challenge to overcome the disparities when there's, you know, this topic expands much beyond science and like into globalization and trade and etc.
Yeah.
Because that is going to determine agricultural practice, which is going to influence the spread of these resistance genes.
That is such a good point that I hadn't thought about what you said in terms of the sanitation practices and how that's a really big indicator for antibiotic resistant genes prevalence. So yeah, as you said, this topic does tie into so many different sectors like globalization, but also agriculture, sanitation, socioeconomic differences, and how we do live in a very globalized world. So it doesn't matter if in this corner of the earth we have all of these regulations if, you know, the situation isn't the same everywhere.
Yeah, exactly. Also, if you just think about, like, animals are a crazy medium to be spreading these resistance genes because they do spread their feces everywhere as well, and including in soil. And I read that there have been experiments where they fluorescently labeled E. coli cells and added them to soil and found that they were able to rapidly acquire resistance genes from the soil microbiome, which is just one example of how, for example, you could be feeding an animal antibiotics, then they could excrete it into soil. Soil bacteria could take that up. That could maybe then be found on our carrots, and then spread into humans, and then humans produce feces, which goes mainly into wastewater, which is another major super spreader of these antibiotic resistance genes, water.
Yeah. And speaking of waste management and how antibiotic genes get into our bodies, I also wanted to point out that the way that you discard your antibiotics is also very important. So if for some reason you ended up not taking your antibiotics, or, well, firstly, you need to take the full course if you do get them prescribed and you do need them, because if you don't and if you feel like you're getting better, the symptoms may have improved, but you haven't completely eradicated potentially all of the bad bacteria. And if you stop halfway, that's leaving the door open for them to become more resistant, and the bacteria that survived up until that point to then share their DNA. But also, if for some reason, I don't know, you have antibiotics that are expired or that you didn't use at home, you can always just go to a pharmacy and hand them in if you didn't know how to dispose of them. In the UK, that's something that you can do. But I also wanted to point out that it's not all doom and gloom. So there has been research done into how to overcome the antibiotic resistance, and I wanted to talk about a few of the methods that are being used. So viruses and bacteria have had a very long ongoing battle where they try to eat each other or gain dominance over each other. And so that is something that scientists are tapping into. So they're using, in some rare cases, phage therapy. Phage therapy is used as a replacement for antibiotics in the cases that are required. So phages are viruses that target bacteria, and what they do is they inject their genetic material into the host bacteria, self-replicate, and then release their progeny through bacteriolysis. And what's cool about phages is actually they can be really specific, so they can just target the exact bacteria that's making you ill rather than targeting like a whole host or a whole spread of bacteria types. Sometimes when you take antibiotics, you get like a flora imbalance because you've just killed a bunch of bacteria that were actually good. And so with phages...
Which is why you should always take probiotics with your antibiotics.
Yes. And so, yeah, with phages, it's cool because you can be really specific. It also helps with things like toxicity, and as mentioned, it minimizes perturbations to normal flora.
Wait, sorry.
Yeah.
Could you explain how exactly you can be specific?
Yes. Phages can be specific because firstly, you can deliver them to the site of the infection. And secondly, they bind to specific receptors on the bacteria's surface. And phage genomics can be manipulated and edited in vitro before they are introduced into their bacterial hosts, which, yes, do allow you to be very specific with what bacteria you target. So it's interesting, again, talking about the regulation of different countries, because in the US, it's not FDA approved except for this one exceptional case where it was required. And I think their stance is it's not approved, but in extremely exceptional cases, like they make an exception. In the EU and in the UK, there have been some phage therapy trials and treatments, so it's a little bit more open. But in Eastern European countries like Georgia, Russia, Uzbekistan, and Ukraine, it's actually quite commonly used. And I think it's simply due to history and geography, because penicillin was discovered in the West, whereas phage therapies were just taken up in the East, and that's been the standard. And I don't know why it's taken so long for the West to use phage therapy, and now it's becoming more common due to the antibiotic resistance, and so we require phages now.
Interesting. I wonder if phage therapy though could have any downstream effects, like if they're modifying bacterial DNA, that has to have like a very specific desired effect. Basically, I wonder if there could be any like downstream negative effects of that, because if it's not like controlled, then you could also be modifying the genome of bacterial DNA in a way that could maybe have downstream effects.
I guess potentially, and I guess that's why they're still carrying out clinical trials and assessing what is the best method and what the side effects are. But something that is also quite advantageous to using phage therapies is that it has an anti-biofilm activity. So biofilms are what happens when clusters of bacteria are bound together by polymeric substances. And this polymeric network essentially connects and immobilizes bacteria cells within the biofilm, and they act as barricades from physical, chemical, and biological challenges. So if you have a really severe bacterial infection and you develop these biofilms, it makes it extremely difficult for antibiotics to get through. And because it allows for these bacterial cells to be less metabolically active and essentially live for longer, and they're not affected by antibiotics. And phages can penetrate and diffuse through water channel networks within the biofilms.
Wait, so that's really cool. So are they together with phages, or is it a whole separate therapy?
No. Phages do this. The phages can do this.
Oh.
Yes.
So they can have an antibiofilm effect.
Yeah.
Oh, okay. Yeah, another interesting idea would be to have basically targeted antibiotics so that they could be coupled with like an antibody that then, doesn't affect the whole body, but just affects,
Yeah.
Were you going to expand on that?
No, I was just going to say those are the two other methods that are used, immunotherapy and monoclonal antibodies. Yeah, it's really cool. So immunotherapy involves the use of antibody-based therapies, and they've traditionally been used in cancer therapy, but it has been shown to be able to be used for treatment of bacterial infections. So essentially, bacteria and bacterial proteins can function as non-specific immune activators that trigger immune cells such as monocytes and macrophages. And so when an antigen is recognized, it stimulates T helper cells and B lymphocytes, which are white blood cells that produce antibodies and that lead to bacterial opsonization. So they eradicate the bacteria. However, there are really large challenges with using immunotherapy because it has a really high manufacturing cost and regulatory approval. A similar type of therapy is monoclonal antibodies, which again have traditionally been used for a different type of disease, usually rheumatic disease and also cancer. But now they're also starting to use this monoclonal antibody treatment for bacterial infections. So the way that it works is these monoclonal antibodies bind to key scaffolding proteins in bacteria, which disrupts the integrity of the biofilms from these bacteria. So essentially this molecule can cause the collapse of bacterial biofilms and leaving the bacteria vulnerable to antibiotic therapy. So I think that oftentimes it's used in conjunction with antibiotics. And even though all of these new therapies are coming about, it's also kind of interesting that there isn't a lot of funding going into this, especially from pharma companies who you would have thought would be the first to jump on, new drugs, more money, but actually they're not doing that due to complex intellectual property issues, as well as a lot of regulation and development and making of the drugs, as well as the fact that the return rate isn't as high. So there's less money going into this, which is also a little bit of an issue because we do need these therapies to come into light and to undergo clinical trials. So yeah.
What I find really interesting is, I like that there are these alternative therapies being developed, and that a lot of them basically rely on more localized effects rather than in your whole body. And if you think about it, antibiotics would inevitably affect your gut microbiome a lot because you're ingesting them. And to think that if we have therapies that really target the infection site, which if you have a bacterial infection in anywhere other than your gut, it wouldn't affect your microbiome. How would that change or maybe improve human gut microbiomes,
Yeah, definitely. That is something that I agree with you is quite exciting about these alternative therapies, because commonly when you take antibiotics you then get thrush, and then you then need to take antifungal, and we didn't get into fungal resistance, but that's not a good area either, so
that's true. I guess it does affect other microbiomes as well. But and also from a neuroscience perspective, all I'm thinking is like, what's the neuro effect?
Yeah.
And obviously there's a huge link between the gut microbiome and like mental health. So that's why I was thinking about, if we could have a way where you don't need to take antibiotics and then like get really sad,
Yeah.
then it would be way better.
Yeah, definitely. I think we'll leave it there for today, and I hope that you enjoyed today's episode. I hope that this topic has inspired you to, raise awareness on antimicrobial resistance, but also understand and appreciate the complexity of not only the biology, but also the socioeconomic structures that are at play, the agricultural structures, the policies, the regulatory bodies, and just know that you're part of this whole big system and that you can do something to help.
Yeah, I think antibiotic resistance as a topic has been a huge reminder that humans are part of the greater ecosystem, and that our human activity is can have such a big effect on the evolution of other species, such as bacteria. And so yeah, I definitely echo that on, of course, antibiotics have been extremely useful for the history of human health, but we also need to harness our power to be innovative, to not let that effect become a negative one, and keep it positive.
We hope you enjoyed today's episode. Please feel free to check us out on Instagram at neuroverse_pod and our website neuroversepod.com for more information. Thanks for listening. We'd like to thank the Biochemical Society for supporting Neuroverse, a podcast about science and philosophy. This episode is part of our series to disseminate research done in biochemistry.